Okay, good afternoon. This somewhat cloudy Friday. It's time for the Friday seminar, 1 o'clock seminar again, and to our great pleasure, we have the opportunity to listen to one of the few persons that has experience in handling high pathogenic viruses. Viruses that only can be handled at the highest security level by safety level 4. And this is Associate Professor Mirazimi. He has been working and also leading the work many years previously at the high security facility at Stockholm, and also be one of the persons responsible for setting up diagnostics against Ebola infections here in Sweden. Because even though we don't have the virus circulating here, we need to be prepared to, if there are people coming here with suspected Ebola infections, they can be taken care of in Linköping University Hospital, and the analysis is made by this high security facility. And so Oli is one of the few in Sweden with the experience handling these kind of bugs, and we're looking very much forward to listen to your talk. Okay.
So, thank you, Michael. So my lecture will, as Michael said, focus on Ebola virus. So I will give a quite short introduction of this virus family. I will touch the virology point of view and maybe pathogenesis. And then I will shift it to the situation in Africa at the moment with some updates. And end it with the preparedness we have in Sweden regarding this kind of diseases. So, Ebola virus belongs to the family of viral hemorrhagic fever viruses. As it's telling you, is the disease actually manifested by hemorrhage, which principally is causing a multi-system syndrome. So several of your organs, when you are affected of the virus, is out of the function. We also know that the virus affects the vascular system, so your vessels cannot regulate regulate itself, and this is also very important. And we also should mention that this disease is caused hemorrhagic fever or biothreat. There are no medicine, there are no antivirals, there is at the moment no vaccines against them. Why they are very dangerous. And most of them is also highly pathogenic, as most people get the disease, die. So, which viruses cause hemorrhagic fever? There are 4 families. One of them belongs to the family arenavirus, and for this family, we know at least the most famous one is the Lassa fever, which is endemic in West Africa, actually where we have now this Ebola. So otherwise they have usually Lassa fever. Other members of the arenavirus is the New World arenavirus, which you can find it in South America at Champre and others. The next family is the filovirus, and filovirus, this is the subject of today's talk, is the Ebola and very close, call it sister or brother of Ebola, which is Marburg virus. Then we have Bunya viruses, and this is one of the biggest virus family we have, more than 350 members. The dangerous one in this family is the Crimean Congo hemorrhagic fever, which actually you find it from Eastern Asia until the Eastern Europe, and even Southern Europe, actually. And the last one is, of course, flaviviruses, and for this one, you know the dengue. But the dengue is not as dangerous as the other members I mentioned. What is the feature of these viruses? What we know, all these viruses, the genome composed of RNA. So they are RNA viruses. And also all of them are enveloped. So what you can conclude that they are RNA viruses means that these viruses are, as the RNA viruses is quite easy to evaluate, do the evolution. So these viruses easily can adapt to the situation. And you should have it in mind that these viruses are zoonosis. So they have different species to infect and grow. Why this RNA is quite nice for them. They are all envelope viruses, which is for us is good because we know that we can easily kill these viruses. You need only detergent or alcohol, so you can kill these viruses. As I mentioned, there are zoonoses. So human is not the host. So the virus actually in the nature lives in other animals. For Ebola, we believe that it is bats, fruit bats. I will show you some pictures. And also one other important feature is that these viruses are geographically very restricted, except the Crimean Congo, which we know affect 40 countries in the world, but most of them are very focused. For example, Ebola, previously we thought that it was only in the Central and Eastern Africa. Now we know that the virus even exists in the West. But for the Lassa, we know that it only exists in the West Africa. New arenavirus in South America. So they are very restricted. And this is also most probably due to that they need their own host, which is some kind of the animal or whatever, and they need some kind of the environment. Climate should benefit for this species to come close to each other. So these are the factors can affect why they are geographically restricted. So Ebola. So the virus actually, as I mentioned, this is a zoonosis, and the virus actually get its name because the first time we discovered the virus was in 1976, I believe. It was close to a river of Ebola, in that time Zaire, now it's Congo. And the virus actually affected several villages around this Ebola river and caused, I believe, it's about 400 deaths, which is compared to the new one is nothing. Have it in mind. And this is just a picture showing the river. Ebola virus today we know that there are 5 types of Ebola virus. The first one, which we discovered in 1976, called Ebola Zaire because it was found in the Zaire. But now we have decided to change the name of this virus from Ebola Zaire to Ebola, just to make it difficult how to talk. So the Ebola Zaire is now Ebola. And then we have the other 4 members, which are Ebola Sudan, which of course find first time in Sudan. Then we have Thai forest, previously called Ivory Coast. And then we have the last one found in 2007 is Bundibugyo, which found in the area between the Uganda and Congo, in the area of Bundibugyo. We get the name of Bundibugyo. And the 5th one or 4th one, depends on how you put it. But the one actually does not cause disease in human, called Ebola reston. And the first time they discovered it, it was in Virginia, in US. And this was most due to that they imported monkeys from the Philippines island, and then found that these monkeys died after a while, and then they started look for whatever these monkeys could have, and they found Ebola in these monkeys. They become very afraid what's happened. They checked the staff dealing with this monkey and found that these people actually had antibodies against Ebola. And this same outbreak happened even in Italy and in former Yugoslavia. The same situation. Then actually they look back, they try to find where the monkeys comes. They found the monkeys come from the Philippines. And they go to the Philippines and they found even swine get the disease, this Ebola reston. They show maybe some kind of the very mild symptoms, respiratory symptoms. But the people working with the swine also had antibodies. So we know that this reston actually is not pathogenic for human, but kill monkeys. And sorry for the Swedish one. This is the fruit bats. And these are the guys. I don't try to spell the name, but they are quite awful one. where you find this fruit bats. This is the area in the red. You see this is where you can find these fruit bats. We believe that these fruit bats could be the host of the Ebola. So I will now show you the picture where we find the Ebola. So have it in mind, this picture, and then we check this one. And this is where we see the Ebola outbreaks. So it is quite nicely in the same spots, actually, where we have the Ebola and we find these bats. The different colors showing different strain of the virus. So the red one is the Ebola Zaire, formerly Ebola Zaire, now there's Ebola. The green one is the Sudan, and the blue one is the newest one from 2007, Bundibugyo. And here the small one is the Thai forest, or Ivory Coast. And the current outbreak is this one. Again, we believe that it is belong to the same strain as Ebola Zaire or Ebola in the West, which is quite, this area never showed any Ebola. And this picture, the next one, just showing you where this Ebola restaurant found in these Philippine islands. And I most probably, here they're talking 4 human was that this is the data from the American outbreaks, but they go back to the Philippines and find even the people in the island had antibodies against the virus. So, what is the clinical presentation? So this one you have, all of you have heard it in the newspapers, TV reports, whatever. So we know that actually the symptoms, the early symptoms is very diffuse. So it is as you get flu, fever, suddenly fever, you get a headache, you have after a while muscle pain, and then lately develop to the pain in your stomach, diarrhea is the one, and vomiting also. These actually the characteristics that they follow by very diffuse and end up with this. And I just heard to the 2 doctors coming back from Guinea, and they told me about this patient. One of the biggest symptoms they see in the patient was that they were so tired, these people who get the disease. And very young, very strong boys, men, actually couldn't stand on their foot, on the bed, because they were so tired. So and usually when we're talking about Ebola, usually you see in front of you people bleeding from all over the places, but that's not true, for sure. At the bleeding stage is the very latest stage, and not everybody showing this bleeding stage from the skin. So what you do is you see some bleeding manifestation from the mucosal area, from nose or from the mouth, whatever. We're talking about the incubation time, and there is a different incubation time. WHO say 2 to 21 days, but most of the cases we know that happen within a few days up till 10 days. But from the WHO side, try to be in the short side, so they choose 2 to 21. By this, we can really restrict the spread of the virus. When we're talking about the mortality of Ebola virus, so you have seen, I will show some picture about that. You see some outbreaks show up to the 90% mortality. Some show, I mean, if you remember for a few months ago, people talk about 50% mortality of this new current Ebola disease. Now they say that it is 70%. However, without discussion about why that's one we can discuss later, why we see this discrepancy between the 100 and 50. But as you may understand, there should be some differences. What is the difference? Why some people die? Why some people survive? But what we know is that when we look at the people who study people dying and surviving, what we find is that those who die at least have 100 times more viruses in their body. We know that. We know also that both of the cases, survivor and non-survivors, respond by IgM. So there is an IgM response. But the non-survivor, never develop IgG. And this could be quite clear reason that because these people don't survive more than 2 weeks, and you need 2 weeks to produce IgG antibodies. But IgG response could really be a good prognostic diagnosis for outcome of the patient. The patient showed a good IgG early, after 5, 6, 7 days, you can be quite evident that this patient can survive the disease. We also know that these patients usually lose lots of leukocytes, most probably due to the apoptosis. This one we also maybe touch. And we also find lots of nitric oxide in the non-survivor patients. Okay, how the virus transmits. This is also something you, if you follow the newspaper, you get a quite nice clue what's happened. So, but we know for sure that to get the disease, you need to be very close to a sick person. And the sick person, what we mean is that the person showing really Ebola symptoms. And with the contact, we mean that the body fluid contact. So discuss with these MSF doctors, they say that in the hospital, they have 1 meter's distance between the patients and the family. And they try to skip the body contacts, because as soon as you have body contacts, you will have some body fluid contacts. So body fluid is definitely could be the reason of transmission. One other big aspect is this one, and it was a paper published in Science I believe it was in the early September this year, actually demonstrating that starting of outbreak in Sierra Leone depended only due to one of these ceremonies. That when one healer, because you know in Africa the different villages, they have different beliefs. In one of these villages there was a healer, and she thought that she's the person who can heal the Ebola patients. So he healed lots of Ebola patients, which you may understand she never managed. And at the end she get the disease and she died. But for the ceremony also participate 2 other healers, 12 other healers. And during the ceremony they have usually the cultural stuff is that they should touch and kiss the dead body. So these 12 actually get Ebola during this ceremony. They did go back home to their own villages and they start to spread it. So within a few weeks, Ebola started in Sierra Leone and distributed hugely in the rest of the in the rest of the in the rest of the Sierra Leone. So this is a very big important issue for WHO and for MSF. Try to have this kind of the handling of the bodies. So in the beginning, it was very big problem for the people in Africa trust to the authorities and hospital, because you can have it in mind in the hospitals they call it actually African people. I have a friend actually from Africa. He told me that we say go in the box and come out in the box. So this is what they see. They go to an hospital. They don't see the family when it's go to the hospital, and then after 2 weeks, the member of family comes in such boxes out. So they lose the contact, and this is known by MSF. So now in the new hospitals, they try to have very open areas, so they can actually relatives can be in other sides and see their sick members, and they try to enjoy this ceremony by staying around to have a clue, but not touching. So this has been a very big issue for this outbreak and the previous outbreak as well. So this is just showing you, comparing 2 other members of this hemorrhagic fever virus, Lassa fever. You see that 20% is symptomatic, so 80% could be asymptomatic, and the same is for Crimean Congo. But for Ebola, most of the patients demonstrate the symptoms, which means that they develop the disease. And this is clearly because this virus is not belong to us. This is an animal virus. However, monkeys get also disease. How virus look like? This is some electron microscopy picture of the virus. You see that the virus can have a different shape, of course, depending on the media. If it is in the culture media, if it is from saliva, it is directly from serum, or in the different organs. So the form of the virus can be different, but it is filamentous virus quite nicely. And we don't know why, actually. Sometimes they can be very, very long. This is actually a big area for research to understand why they are so long and some so short. So a schematic picture of the virus look like this. You have this glycoprotein outside of the virus. This is the enveloped virus as other hemorrhagic fever virus, means that you can easily kill the virus with alcohol or detergent. The virus contains actually RNA, if you remember. It is a negative polarity, the RNA, and it is a single-stranded RNA. Protected, I will show some pictures just demonstrating. So the RNA of the genome of this virus codes for 7 proteins, actually. And one of them is glycoprotein, which is a surface protein. During the infection, processed to 2 glycoproteins that make actually the outer shell protein of this virus, which is important for attachment, finding new host cells. So just, this is a more very detailed, I don't need to go through all of this issue, but so these different proteins have a different function. We just skip this. But what is important is that this virus is coded for their own polymerase. And this is very important. And this is showing you the replication cycle of a virus into mammalian cells. So as other viruses, it enters to the cells, produce new viruses, and buds out from the plasma membrane. It's not a big issue. By this picture, I try to demonstrate the receptors, because the most important is the receptor in the term of the therapies. Have it in mind that this virus, actually, if you remember what I said in the beginning, they can infect different animal kinds. Also, in the body, they can infect almost all cell types, which means that this virus actually has several different receptors. And this is what I'm showing here. We know that they are attaching to this different kind of the lectins and different of kind of the integrins. And lately, it was for 2 years ago, maybe it was, this paper was in Nature, demonstrated that the virus also needs this Niemann peak, which you find it in the endosomes. And we believe there are many other receptors also, because the virus can infect bats, they can infect monkeys, they can infect swine. We know that there is antibody in the dogs. So most probably this virus has several different receptors, which is important for understanding the pathogenesis and also the tropism of disease. I will come back to this picture and discuss other aspects. This is just showing the structure of the glycoproteins. And glycoprotein is one of the most important structure of this virus, because if you can neutralize the glycoprotein by antibody, you can actually stop the infection. So the virus codes for only one glycoprotein, which during the replication processed to 2 glycoproteins. And these 2 proteins, or 2 part proteins, build 3 heterodimer molecules, which actually find all the receptors I discussed on the different cell types. And this is just showing you this picture. So when we get this is the real glycoprotein here down, it will be cleaved here and make 2 G1 and G2, then the virus actually managed to produce half of the protein, which is read only by orphan. This is very interesting because this protein actually secretes from infected cells. So if you think that if your body produces lots of neutralizing antibody against the glycoprotein, which usually should be only on the envelope of the infected cells or on the free viruses. But if you have free secreted glycoprotein, this protein can actually attach to this neutralizing antibody, and by this escape the immune responses. So this is one strategy of the virus to escape the immune response of the body. Quite clever. And this is just showing you this. This is the glycoproteins. So these are those I told you. They go out and find the antibody and block the antibodies. By this, the virus can actually escape the immune responses. The pathogenesis. What we know about the pathogenesis. What we know that the patients who die, they have not good, how to say, cell-adapted or humoral immune responses. They have no T-cell responses and they have no IgG responses. That's we know. We also know that the lymphoid cells go to apoptosis. This is also what we know. We also know that dendritic cells also go to apoptosis. How the virus managed to make this. What we also know is that these viruses are very sensitive to innate immune response. So just make an easy example. If you take a cell in a culture cells, put interferons, which is one of our innate immune responses, put the interferon on this cell, then put the virus can never ever grow in these cells. Because interferon is has a very high antiviral activity against the virus. Then you can ask me directly, okay, if you have this antiviral activity of interferon, why the body cannot combat the infection? The answer is, we believe that we have the answer. One of the protein codes by this virus, VP35, is an antagonist. So this protein can, by different way, stop the production of interferon. So the virus, as soon as it comes, start to produce VP35, which can dysregulate the production of interferons, which is normally introduced as soon as something, virus or bacteria come in our body. By this, the virus have time to grow. We also know that the virus, by different mechanisms, can actually stop the maturation of the T cell and B cells. I will show a picture about that as well. So putting this together, so what I am showing here, this glycoprotein, if you remember this half glycoprotein, they can mask the neutralizing antibody. By different proteins function, we can stop the innate immune response, the very first innate immune response. By this week, the virus have time to grow and establish the infection. So if I put all this together, so I can show you this picture. So this virus actually by infection first infect dendritic cells. That we know. This is how the virus works. Infect the dendritic cells. The dendritic cells go through the blood vessels, blood system, to the lymphoid organs, to the spleen, to liver, where they actually meet other cells. In the normal infection, dendritic cells, when they are infected, they present the protein of the virus, go to the spleen, present it to the other lymphoid cells. Then we actually induce lots of T cells, B cell response. In this case, we have learned that when Ebola infects dendritic cells, actually the virus managed to stop the maturation of dendritic cells. What's happened, the dendritic cells, they don't show the virus protein on their membrane, but they actually spread the virus. So what they do is they take the virus to the different organ and present it to the macrophages, other monocytes. And when these cells get infected, they start to produce lots of cytokines. So what's happened is that you have a very local pro-inflammatory response, which affect lots of the function of the organs wherever you have. This can be in the liver, it can be in the spleen, it can be in the kidney, whatever. So this actually damages the blood vessels. Why the blood vessels cannot regulate and become leaky. At the same time, we know that the virus can also affect the coagulation factor, which lead that you bleeding, but you cannot stop the bleeding. We know also that the virus can infect endothelial cells. Endothelial cells, the cells actually build the vessels. They also affected, and the effect of this is that completely you lose this regulation function of the vessels, which lead to the shocks. So what's happened? You destroy your liver, which is very, very important. You destroy different other lymph organs, and also in different other gastroenterial organs, and they lead to the diarrhea. So what's happened is the circulation failure, and then later on you lose the regulation of your blood pressure, and this leads that the patient die. This just tried to describe that when we're talking about this losing the tightness in the blood vessels. If you think that this is the blood vessel, these are the endothelial cells building the blood vessels. So when the virus infects these monocytes, they produce lots of cytokines. These cytokines actually affect the expression of integrins, which lead that this decrease. What it in turn means is that these cells actually lose the junctions. So that's what actually happened. So they lose the junction and they start the leakage. As I mentioned also that previously we had S here means that soluble, also half protein which are secreted out from the cell. This protein can also interact with the neutrophils, which are very important in early infection, and also stop the activation of these cells, which is quite important. This one we have discussed it a lot, so I don't go into the details. This picture showing, and by this one I will summarize the most part I discussed during these few minutes. That Ebola virus infects dendritic cells. Dendritic cells never maturate, which means that they cannot activate the T cell and B cell, but they actually can induce the apoptosis. So these cells go to apoptosis. Maybe I'll show a picture how the mechanism of this, but whatever, these endothelial cells lead to the apoptosis of T cell, B cell. You never get any adapted immune response, which means that the virus can grow. At the same time, endothelial cells, dendritic cells, can lead to infection of macrophages, other monocytes, and go to the different organs, especially we talk about in the liver, hepatocytes, which are very important, make these cells die by apoptosis and produce lots of virus. Lots of cytokines, if you remember, cytokine storms lead to the leakage, virus and hemorrhage, and stop the virus inhibit the activation of NK cells and neutrophils, most probably due to this soluble glycoprotein, and no response of this. So what's happened, the virus can easily escape the immune response, produce lots of virus, and survive. This one, I believe I touched it more or less. So by this, can make it quite easy summary. What virus do is that inhibit the early response, innate immune response, interference, if you remember, this is most important. The virus manage also inhibit or delay the antibody response. In that case, I mean IgG response, and also T cell response, which lead to the uncontrolled viral replication and at the end, death. Okay, this was by here I will show you, because this protein B7H1 is very important for induction of apoptosis. By interaction between this protein and the receptor in the T cells, you can induce the apoptosis. But I believe we skip this. And this one we skip. But this is interesting. This one, so you, as you may understand, this is quite a virus, it only has 7 proteins, not more. Okay, the other viruses has only 3, as Crimean Congo. But this has 7 proteins, comparing to the herpes viruses, which have 250 proteins. So they are quite easy. So you may say that, okay, it should be very easy to make vaccines against this virus. Most probably it is. And this showing all the vaccine research has done in only in US. And you may ask, why US? The answer is easy, because Americans have lots of soldiers and whatever people around the world. So they are very interested of this disease is caused that. So they have, this is mostly in the military, army defense programs. They developed several vaccines. I marked the red one as the good one. This study based on the mice, small animal. So you see that they have quite good, the red one has a quite good survival. So they could protect the mice from dying by 100%, most of them, during the last 5-10 years. Then after that, they go to the monkeys, because monkey as humans show the disease. And this is just showing the study done in the monkeys during last maybe 5 years. So there are, here I marked 3 of them, which are quite big interest at the moment also. One is the virus-like particles, which is very easy to produce for the Ebola. Actually, you need 3 proteins. Put these 3 proteins in a one cell, you will get virus-like protein. Very, very easy. And they show that by vaccinating monkeys, they could get a nice survival up to 100%. The other one is the recombinant adenovirus, which is the one SmithKline using for the vaccine trials now in Africa. This protein expressing glycoprotein of the Ebola, they show also 100% protection. The other one is developed by Heinz Feldman in Winnipeg. This is a vesicular stomatitis virus, is a close member of rabies. They also show nicely 100% protection. So there are 2 of these viruses, two of these platforms using for the clinical trials at the moment in Africa. One is the adenovirus, the other is the VSV. So let's see which one is the can get the market. And there are several other antivirals. I will not mention them. There is actually one very promising, which is T705 or Favipiravir, I believe is the product name. Actually is a nucleoside analog, which is actually developed for the flu. And in Japan, they have used it for treatment of the flu, and it is in the phase 2 trials. But they tried it for the Ebola and find that it was very nicely could inhibit viral replication in the cell culture and mice model. So they have started a monkey experiment in US. Seems quite nice, and they got I believe 100 million Swedish crowns to do the clinical trials in Africa at the moment. So that's also one of the promising antivirals. Let's go to what is the situation at the moment. I realized that these are old pictures. Okay, this is the new one. This is from 21st of November. So it is 1 week. They didn't publish anything yesterday night. So this is for every week new cases. So you see that for every, from the beginning, increase for every week, number of the, here there is 1 lakh, and they believe that there was no report that week. It doesn't mean that the virus was away, it was just they had no reports in their database. And then come a lot here. And here you see a very huge peak. This is not that that week was a lots of cases, it was just several reports come in that week together. So it could be planned out here. So what we see is increasing. Here we see maybe some decrease, maybe. But this is, you cannot be sure about that until a few weeks after. Maybe this collection of the data and they come again here like this. Whoop. So let's see what's happened. So if put it in the map, so the size of this dot showing the number of the cases during the last 21 days. As you see, Liberia and Sierra Leone completely infected. They still have infection ongoing. The number, I talked to one guy who is in the capital city in Liberia, and he told me that the number of the case in the Liberia decreasing. Last week they had 200 patients. This week they had only 30 new. So this is really decreasing in the Liberia, but increasing in the Sierra Leone. So the situation is the same in Sierra Leone, but decreasing in the Liberia. But most interestingly is Guinea here. Here is the area when the virus starts, the epicenter. What we see still is that the spreading ongoing in the first spot. Or still ongoing, because the first transmission occurred December 2013. So now we are close to the 1 year. Still we have transmission. And Guinea, they have quite good infrastructure compared to these two. So we don't know what will happen. If maybe these huge outbreaks go down, but then we maybe will have a really ongoing epidemic in this area for a long, long time. That was a picture from WHO. This is a picture from ECDC. They're more or less demonstrating the same thing, that the situation is still very, very serious. Let's see what this one's showing you. Yes, this one is interesting because this showing the number of transmission in the hospital to the doctor and nurses. So this is the percent of the infected, and this is the percent of the death in total point of view. So you see about 5 or 6% of those dying, 5-6% get the infection in the hospitals. And this is showing you here is the West Africa, and you see there are lots of pills, arrows to the coming to the Europe and also to US. This showing those who going to Africa to help, but get the disease and come back home. So these arrows showing those who come back home and had the Ebola. This is just, I try to really describe each cases. This person get the disease or evacuated from Sierra Leone to the Hamburg. He was an epidemiologist. He recovered. It's confirmed that it was actually Ebola, and he was a Senegalese. So here you see that a few of them. This is, I believe, 3 or 4 weeks old. So there are many more. A few dead of this, I believe, a few is dead, and a few is discharged. And you see, we have Dutch, we have US, we have Spanish, we have one Norwegian one. You may remember. Few from Sudan, US, and even Chinese. They are there. This picture, I just This one I have already mentioned. That was the starting of a spreading of Ebola in the Sierra Leone during this ceremony, and started here, and then it spread to the rest of the Sierra Leone during just one ceremony. During this study, actually, which was published in Science, they could do the dendrogram and try to understand when the virus was established, and they found that actually the virus was established in 2000, I believe in 2004. So they said that this virus has been around for 10 years, but we have not seen the outbreak. So the virus has been in Africa. So this is one hypothesis. Let's see the more hypotheses. I will actually take two examples demonstrating the situation we had first was in Singapore. This is just a practical examples that I usually take to show you how the virus can spread. They found a very young man come to the Senegal. Actually, there is why my friend is running this pastor lab. He told me that this guy, they thought in the beginning it was malaria. He had all the symptoms. He get treatment. He did go back home to the village around where he lived with his relatives. After a few days, he come back again. He never recovered from the disease. Later on, actually, they get contacted from the Guinea. In the Guinea, they called to the Amadou, who's the guy in Pastor. I said that actually we believe that a very young man escaped the isolation unit in Guinea because he was afraid that he had Ebola. But he said, if I go to the Senegal, maybe I get a better healthcare. So he he get Ebola, actually, but there was no more transmission. The other one, which is also quite interesting, is the first case in Nigeria. In that case, we know that one financial guy fly from, I believe it was from Liberia, through several stops before end up to the Senegal. During one of these stops, he come in contact with a guy, actually transmit this guy. When, however, this financial guy come to the Nigeria, he get Ebola, it demonstrates Ebola, they put it in isolation, he died. They tried to control everybody and it was no problem. But the other guy he actually met during one of these stops also go back home, some other city in the Nigeria. This guy gets sick after a few days, but it's flu. Go to his job and start to work. But this guy actually was a medic. So you can think what his job was daily, meet lots of patients. So he managed during these few days transmit lots of patients and several colleagues, doc and the nurses. Then we had an outbreak in that area with 2021. So this is also, but they managed actually to stop that, and now there is no more cases in Nigeria. But other side, we start to see some things happen in Mali. And Mali is quite still is in the good part of the Mali, if there is any good part. But that is less, if you go up to the north, then is the war, and that's a big issue. So hopefully they manage to handle the situation. So this is just showing you how the transmission happened. This is just a picture showing almost all the outbreak we had from 76 until a few of them, and the last one I didn't bring here. So you see the number of the transmission case and number of death. So you see, as the biggest number you get is 400, something around. Mortality differs from 80-90% to maybe 70. There are some is 100%, but don't bother about this because there is only one case. So this doesn't tell you. But we see the number is usually 200, 300, 400. But this time we are talking about, WHO say about 15, 16, 17,000, which is reported. And most probably twice is not reported. So we are up to maybe 30, 40,000 affected. So it is not comparable. Then we can discuss why this, compared all this. Okay, let's end the talk to see what can we Sweden do if Ebola comes here. So this is a picture showing the facility in the Linköping University Hospital. Actually, they can take care of, as I understand, 3 patients at the same time. So we cannot have a bigger outbreak here. 3, only 3. But most probably we will never have more than 3 because the virus is not here. Maybe we have one of the docs get the disease, come back home. So it should be good enough. We have also this special ambulance so actually can take care of the patient, move them from Kalmar to Linköping or from Umeå to Linköping. So the facility is quite good and they have quite nice SOPs and send it around to all hospitals. So I believe hospitals know how to handle if such patients come. What we can do when the patients come, we need to do the diagnostic. How we do diagnostic? We do of course, molecular biology, because if you remember, this virus is transmissible when it is showing the symptoms, which means that when you show the symptoms, you have virus in your body fluid. If you have a virus in your body fluid, then you can do the PCR. So it is good enough. There has been some discussion previously, if you get the disease, if you say for the last outbreaks, when you get, because the Ebola is not the first thing you think, so when we get the samples, or the lab get the samples, central lab get the samples, the patient is really sick, means that they have lots of virus. The sensitivity of PCR is not so important. But nowadays, it is good enough if you get the flu symptoms and you have been in the air of the Guinea, if you are sitting in the plant. And they directly say, okay, you maybe have Ebola. Then we can get the samples. So even you have Ebola, the symptoms is so early, so maybe PCR has not the sensitivity. Why the CDC recommended that these patients with a very highly suspected for Ebola should be in isolation for 3 days. And in Sweden, we said this is impossible. It costs a lot for the landstinget. So why we said, oh, maybe 24 hours is good enough. ECDC said 48 hours. So, 24, 48, 72, I don't know. But most depends on the PCR. If you have very sensitive PCR, you don't need to have the isolation. Besides the PCR, we have also can use the IgM and IgG. IgG maybe is not the best one because maybe they don't have IgM. But IgM they have, and usually they show it 2-3 days after. But the problem with the serology is that no serology at the moment is validated because we have no patient sera. Previous outbreak in Africa always connected to CDC. So American come, make the diagnostics, took the samples, go back home. And no European countries seen the samples. So we in the Europe, they are not validated. But now we are in the field, so we have access to samples. We have, of course, electron microscopy, one of the best methods, in my opinion. The problem with electron microscopy, the sensitivity is very low. And then, of course, you can isolate the virus, which is also good, but then you really require the BCL4. Without that, you cannot do that. So by this one, I will say thank you and open for questions.